food, drugs, or ideas, what you consume influences who you become. On the Mind and Matter podcast, we learn together from the best scientists and thinkers alive today about how your mind-body reacts to what you feed it. Before starting Mind and Matter, I spent 10 years in academia doing scientific research. I got a PhD in neuroscience where I focused on neuroendocrinology and the neurobiology of behavior, and before that, I specialized in molecular, developmental, and evolutionary genetics. I use my scientific background to help parse and translate the information that guests share on the podcast. In addition to the podcast, I write long-form written content inspired by the show where I integrate what I've learned across episodes. I also have a free weekly newsletter where I provide you with upcoming guests, share links, and provide commentary on scientific studies and research that I'm reading, and more. Visit mindandmatter.substack.com to find all of my content. Joining me.
Thank you for having me here today.
Can you start off by just telling everyone a little bit about who you are and what you study?
Yes, thank you. So I am currently an assistant professor at the School of Life Sciences here at Arizona State University. And I also run a research lab out of the Neurodegenerative Disease Research Center, which is housed in the Biodesign Institute. And our goal within the center is to identify, one, environmental factors that are playing into disorders like Alzheimer's disease and other forms of dementia, as well as developing therapeutic strategies for disorders like Alzheimer's disease. We also study the tauopathies. So the tauopathies include disorders like frontotemporal dementia, where you have a aberrant protein that accumulates known as the tau protein. And we use a variety of model systems in my lab. So here in Arizona, we have a unique program that allows us to actually use brain tissue and other forms of tissue from donors that donate their body to science that have these various diseases. And we're able to acquire this tissue and look at protein dysregulation, validate whether these specific targets that we're studying are actually present in these cases, and also use cell models and animal models to test our hypotheses. My lab has a variety of different students at different stages of their career. So we have undergraduates as well as graduate students that are pursuing PhDs, as well as postdoctoral fellows that finish their PhD and are getting extra training in regards to specific topics they're interested in, creating a path for them to become independent and future leaders in the field.
All right. Well, we're going to talk about a lot of interesting stuff. You've done some interesting work in the realm of neurodegenerative disease, especially environmental factors and chemicals we're exposed to that might play into the pathophysiology of some of these things. I want to have some discussion of neurodegenerative disease to give people a sense for what that is at a high level, and Alzheimer's disease, including what we know about the pathophysiology and how the thinking has changed and some of the controversies in that field over the last few years before getting into some of your recent work. So I want to start very basic. What is a neurodegenerative disease as opposed to, say, some other kind of psychiatric or neurological disorder?
Right. So that's a great question. So a neurodegenerative disease is going to be a disorder where neurons, which are cells in the brain that are critically important in producing our everyday behaviors. They're essentially what allow us to speak, what allows us to listen, to have be able to perceive visual information in our environment. When those cells degenerate because of some sort of pathology or proteinopathy, in this case, a protein that does not belong within those cells, that is what we classify as neurodegenerative disease. These neurons are atrophying, dying in certain cases, and ultimately causing clinical symptoms that begin to compromise the ability of the individual to navigate their environment, and in many cases require a caregiver to take care of them and allow them to essentially survive in the environment.
Mm-hmm. So proteins start functioning inappropriately, but it's not just that they start functioning in sort of a funny way. They are sick in a way that will eventually kill the actual cell.
Yeah, that is correct. So let's take, for example, Alzheimer's disease, right? Because it's the most common cause leading to dementia. Dementia, as we know, is classified as a decline in intellectual abilities. What you tend to see in Alzheimer's disease is that there's two proteins that are hallmark pathologies, right? And these were actually first identified back by Alois Alzheimer's, a German neurologist who first identified these two pathologies in the brain of a patient known as Auguste Deter, which he was seeing because this patient had symptoms of memory loss as well as sleep disruptions and also hallucinations. In the early 1900s, when this occurred, he was able to actually get the family to agree to look at the brain of Auguste when she passed away, and he identified two proteins that were aberrant and didn't belong in the brain, and this was the amyloid beta plaque and the neurofibrillary tangle. And what we know is that these proteins are a result of changes that occur in within these cells that compromise the function, right? So think about, for example, when we talk about the amyloid pathology, there's a protein known as the amyloid precursor protein that is produced by the amyloid precursor protein gene. And what happens, it's cleaved incorrectly, leading to the production of the amyloid beta plaque, which then begins to compromise the function of neurons communicating. So that's the amyloid beta portion. And then the tau pathology, this other proteinopathy that you see in disorders like Alzheimer's disease and tauopathies, is a result of a protein known as tau that's critically important in neuronal function. But when it begins to go a process that makes it break off its stabilizing components known as microtubules, you tend to see that it aggregates, accumulates, and they can be toxic to the actual cell, ultimately leading to the demise. Now, in addition to those two pathologies in Alzheimer's disease, you also get inflammation, right? So we talk about inflammation. This is essentially molecules that get released by nearby cells known as glia when there's some sort of insult or pathology that you see in the brain. And essentially what you find is that that increase in that inflammation can also harm the neurons themselves because it's chronic, right? Having these inflammatory molecules show up in early phases of these pathologies can be beneficial, but when they're chronically released can actually hurt the neurons themselves. And that ultimately leads to the neurodegeneration.
Mm-hmm. And so the standard story that I'm familiar with that I think has been the basic model for many years, but I think has some weaknesses in it, although I'm not an expert in this area, is the basic idea here that for whatever reason, things in the cell start to go awry. You get the production of this beta amyloid protein. You get the production of this tau protein. These are things that should not be there, at least not in that form. They start to build up, things get tangled in the cell, and there's just sort of gunk. It gets in the way of normal cellular processes in a way that makes the cell sick and eventually dies. Is that the basic idea here? And how tight is the relationship between those things and the clinical presentation of Alzheimer's dementia?
Right. That's this is a complex question, right? So I'm going to break it down and answering all these different components. So yes, the idea in general with Alzheimer's disease is that these pathologies build up in these neurons. These neurons are producing the behaviors that allows us to navigate our environment. But when they aggregate and they compromise the communication between these neurons, ultimately your behavior is affected, right? So when you think, for example, of Alzheimer's disease, you can think about Alzheimer's disease as being a disorder that's first identified by the inability to develop new memories, right? Mm-hmm. And that's something that sometimes many individuals are not aware of. They think, oh, I can't remember, who my grandmother is or my grandfather is. That means I have Alzheimer's. Well, not necessarily. The first typical symptom that you see in case of Alzheimer's disease is the ability to actually develop new memories, right? So if we were having this conversation tomorrow and I asked you the question, what did you do yesterday at around this time, and you're not able to answer or remember, that is typically a sign or a feature of the Alzheimer's disease symptomology that occurs early on. And why is that? Because a lot of the pathology that starts, it's typically in that area of the brain where those memories are formed, which is known as the hippocampus, right?
So people tend to start out having trouble forming new memories, but still retain the long-term memories they've already stored.
Correct. And then as the disease progresses, the pathology is found in other areas of the brain, cortical regions. Those are going to be areas more associated with judgment, decision making, long-term memories. Those get compromised. And ultimately, as a disease progression goes through pretty much every area of the brain, you get overall intellectual abilities that decline to the point where an individual requires a caregiver 24/7.
Mm-hmm.
Yeah.
And so if the idea is things like these beta amyloid plaques and or the tau protein build up, and this is really the cause of Alzheimer's, you would expect a very tight relationship. If you go and autopsy the brains of people who had Alzheimer's, you would expect all the people who had it to have these things, and that the worse they had it, the more of these things that they have. Is that the case, or do we find people that have these plaques but don't have Alzheimer's and vice versa?
That's a great question. If you look at a patient that has clinical symptomologies and is diagnosed with probable Alzheimer's disease, the way you confirm this is at autopsy by confirming that they actually had amyloid beta plaques and tau tangles present in their brain. That's how you get a confirmation this person did in fact have Alzheimer's disease, right? So it's at death. But yes, you bring up a good point. There are cases of individuals that have amyloid beta plaques and are clinically fine. They don't develop clinical symptomologies. Why is that? I actually had this conversation this morning with one of my colleagues that runs a program that's really centered around physical activity for individuals that are over the age of 65. And what I was communicating to him is that depending on the cognitive resilience you have, right, which I'll define what that is, that can actually make certain individuals not be affected as much as individuals that have lower amounts of cognitive resilience. So what is cognitive resilience? It's essentially, think about it as use of your brain in capacities that kind of creates a buffer so these promopathies cannot really affect the cellular mechanisms and ultimately alter the behavior. So engaging in cognitive activities has actually been shown to be one of the mechanisms that can actually lead to protective effects, even in individuals that do show, for example, amyloid beta plaques. So the story is complex, right? Yeah. Because once again, if we look at autopsy and here in our local brain bank, we actually see there's some individuals that do develop amyloid and tau pathology, but are clinically okay. Mm-hmm. So what we're trying to do with those cases is understand, why is that? What changes in those cells do they have that made those neurons resilient, even though the pathologies were actually present?
Mm-hmm. And to the extent that's true, I suppose that would imply that the buildup of these plaques, even in these resilient people that don't present with overt symptoms, that doesn't necessarily mean that the plaques are benign or not doing anything. They simply just might be sort of powering through any extra difficulty, cognitive difficulty these things are causing. It's not necessarily the case that these plaques are totally, just harmless.
Yeah, so there's definitely evidence in the literature, if you look at some of the experiments done in cell models, animal models, showing that the amyloid beta plaques can in fact cause some form of toxicity, right? And can elicit inflammatory responses in the brain that can harm the neurons. But once again, the question is, are those given neurons that actually undergo this pathology or have presence of pathology extracellularly? Because the amyloid beta plaques, I just want to clarify, are typically found outside of the neurons, right?
They're outside.
Whereas the neurofibrillary tau tangles are typically found internally inside the cell, right? So the idea is that it's possible that a neuron, with someone that does have amyloid beta pathology, might just have that resilience to resist any sort of interruption that's brought on by the amyloid beta plaque. And these are some of the studies that are currently ongoing. And that's why it's critically important for us to also examine individuals that reach a certain age and are clinically fine to understand why they were resilient and didn't develop aspects of clinical symptomology. Yeah.
Mm-hmm. And I guess there's an issue of cause versus consequence here. Have you guys pinpointed in the field whether these tangles, these plaques, are these a consequence of something further upstream that's going wrong, or are they the cause of the symptoms of Alzheimer's?
That's a difficult question. There's a lot of controversy in the field, right? There's one side that believes that the amyloid beta plaques are causative of the clinical symptomology, because what they highlight is that the amyloid beta plaques are usually the first promopathy that shows up in Alzheimer's disease. But then as you progress and down the the different pathologies, you then typically get the tau pathology, right? And it's not really until the tau pathology is present that you really find signs of clinical symptomology. What you also find there are synapses are lost, right? So synapses are, you can think about a space between two nearby neurons. They communicate chemically that way, right? And by communicating and strengthening those synapses, you're able to produce memories. You're able to engage in various behaviors. And so when those synapses are lost or are not properly functioning, those behaviors are essentially compromised. So if we talk about the trajectory, it is shown that amyloid typically does occur first, and then you typically have the synaptic loss and then the tau pathology. But it's not typically until the tau pathology is present where individuals do start showing signs of clinical symptoms. Yeah.
I see. So the basic order of operations here seems to be that these beta amyloid plaques outside the cells start to appear. At that point, you may or may not have any symptoms that are detectable. Oftentimes, you won't even have symptoms, especially in more resilient people. Later on, it tends to be that you get these tau protein tangles building up inside of cells. That's typically when you start to see the clinical manifestations of the cognitive issues that people have. So sort of putting that all together, I've seen people sort of point to autopsy data and say like, well, we have plenty of brains where they had all these beta amyloid plaques, but they didn't have Alzheimer's disease. It sounds like what you're saying is basically you were probably seeing them at a relatively early stage, and or they may have been a more resilient person, and they could very well have developed symptoms later on if they had survived.
Correct, because it also depends on how old that individual was, if they were, if we're talking about someone that's 55 years of age, maybe they're still early phases, and they might actually be in, what we call preclinical AD. It's not identified. There's no symptomology. They haven't even reached the level of mild cognitive impairment at that point, right? If they were, but we won't know that because there's no way to know that because the individual has died at that point, right? But I do want to make one point because I think what this points to is that the amyloid, in order to have a diagnosis of Alzheimer's disease, you need to have the presence of amyloid and tau pathology, right? But what we find is that typically the clinical symptomology comes closer to the tau pathology. Other side of that solidifies that the tau pathology really might be more of a driver of those clinical symptomologies is that you have tauopathies, disorders where you don't have amyloid beta plaques, just tau pathology, and those individuals also have clinical symptomology. So in frontotemporal dementia, for example, you find the tau pathology, chronic traumatic encephalopathy, which is a result of traumatic brain injuries. You tend to find the tau pathology that leads to these clinical symptoms, and Pick's disease is another example. So highlighting that the tau pathology might be more indicative of the actual decline in clinical symptomology. Yeah.
And I just want to get really concrete about this tau pathology. When we say tangles in the cell, is that just tau protein aggregating together? Is it microtubules getting twisted together? What is the tangle?
So let me answer that question by describing what tau is and what happens in this process. So the tau protein is, you can think about this within neurons as a stabilizer of railroad tracks, right? So microtubules are essentially these various proteins that are critically important in creating the architecture of the neuron, but also are important in allowing proteins to move from one part of the cell to the other, right? So the neuron has various different compartments, and you need to traffic proteins from one part of the cell to the other to allow the neuron to behave in a certain way and produce given behaviors. And what we find with the tau pathology is that, well, first of all, the tau protein stabilizes those microtubules. So imagine a railroad system and railroad tracks with stabilizers, so those railroad tracks don't, when the train is riding on it, the train doesn't fall off the actual track. The tau protein stabilizes that. However, in Alzheimer's disease and other forms of tauopathies, there's a process where that tau protein undergoes what's called phosphorylation, and specifically hyperphosphorylation. It's known as a post-translational modification where the protein tau, there's addition of phosphate groups that cause it to be removed from the microtubules. But in Alzheimer's disease, it happens in a very fast, accelerated manner to the point where the tau can no longer stabilize the microtubule. So the microtubules disintegrate. They can no longer allow proteins to traffic from one part of the cell to the other. And then that tau protein that's now phosphorylated, hyperphosphorylated, then begins to aggregate ultimately into the neurofibrillary tangle. So you have two problems within that neuron. One, the microtubules are no longer stable. You have axonal transport issues, so you can't move proteins from one part of the cell to the other, which means the cell cannot function properly. And two, you have the aggregation of this toxic form of tau, the neurofibrillary tangle within the actual cell.
I see. So in all cells, you've got microtubules, you've got a cytoskeleton, literally a molecular skeleton that gives the cell its rigidity and its geometry and its form. But also, these are important for trafficking purposes. So you use the analogy of a railway. You can think of the microtubules as being like railroad tracks because proteins literally get shuttled from, say, the nucleus out to some other part of the cell or back and forth or whatever. And this happens in all cells, but this is probably especially important in some sense in neurons because they have this odd shape where they can be very long. And you've got to traffic stuff, say, all the way to the synapse. And I would imagine that the reason that some type of pathology like this, a disintegration of the railroad tracks and these tangles based on this hyperphosphorylated tau protein, that manifests in cognitive issues like memory issues, because one of the ways that memories form, an important process, is the shuttling of proteins back and forth to the synapses so that they can be strengthened or weakened or whatever. And so naturally, if you get these tangles and the microtubules can't, the tracks can't be laid down right, then you're not going to be able to engage in the types of synaptic plasticity that our cognition relies on.
Yeah, that's 100% correct. Thank you for summarizing that. And you can also think about it as the synapses and ultimately the what we call the postsynaptic cell, right? That is the neuron on the receiving end. So you have a presynaptic cell and a postsynaptic cell. The postsynaptic cell has the ability to upregulate the number of receptors that the presynaptic cell releases what's called the ligand and binds to those receptors to activate it, right? It relies on axonal transport to do that, to build those proteins that are necessary to increase what are called dendritic spines. And when the axons are no longer able to do that because microtubules have disintegrated and the proteins can not move from one part cell to the other, then you ultimately get compromisation of synaptic plasticity and the ultimately the inability to reinforce certain behaviors such as developing memory. Yeah.
And so if we just sort of move up the causal chain here, you said it was hyperphosphorylated tau. So the tau protein is normally there to stabilize. It's supposed to be there. It's to stabilize these microtubules and enable all of that trafficking and stuff to happen. It becomes hyperphosphorylated in some cases. And this is what sort of initiates the problems we were just talking about. What is the origin of the hyperphosphorylation of tau protein? Do we know that?
Yes, we do know that. So phosphorylation of tau proteins and various other proteins occurs through kinases, right? These enzymes that add the phosphate groups. What we have found, which ties it back to actually the amyloid beta hypothesis, is that the amyloid beta triggers a lot of these kinases to upregulate that then are directly phosphorylating the actual tau protein. So you get more amyloid beta, an increase in certain kinases, and here's some examples. One of them is known as GSK3 beta. The other one is CDK5. One that we study in my lab is known as DYRK1A. The activity of those kinases goes up, which means that the protein itself is more prone to being phosphorylated. And that is why you get the hyperphosphorylation that actually occurs.
And are those kinases specific for tau protein, or do they just kind of phosphorylate a bunch of stuff?
They can phosphorylate other proteins as well, but they tend to adhere or have a predisposition to the tau protein, yes.
And so I have not followed it super closely, but I've followed it a little bit. Over the last, I don't know, several years, some number of years, there have been some controversies and some high-profile papers that were very instrumental in bringing this sort of hypothesis to where it's at today that were retracted, and there seems to be some controversy there. What exactly is going on there, and how has this general theory been impacted by that?
Yeah. Well, so there have been a lot of, in the last couple of years, and I do want to say this is a result, a lot of the development of AI, being able to screen manuscripts and see if there's some alterations in images, which unfortunately are showing up. there have been publications that have been pulled and retracted because the data is not reliable, right? And some of them have been tied to the amyloid hypothesis, right? But the evidence the evidence that points that amyloid beta is definitely present in Alzheimer's disease is still there. There's many papers that support that, right?
Mm-hmm.
So a lot of the papers that have been retracted in the last couple of years that are tied to that are tied to various, forms of amyloid beta, et cetera. But the general premise tied to the amyloid hypothesis that it is present in Alzheimer's disease and it could actually initiate a lot of these kinases or activate them that lead to tau is still sound. So the amyloid hypothesis still has a lot of evidence to back it. Yeah.
Mm-hmm. And if I'm understanding you correctly, sort of my read of what you've said in this whole thing is that beta amyloid plaques tend to precede the tauopathy that seems to be particularly important to Alzheimer's. But what causes the tauopathy is increased activity of these kinases that hyperphosphorylate the tau protein. And one possible initiating factor of that kinase activity going up is amyloid beta plaques. But I would imagine logically speaking, that's probably not the only way that can happen. There might be alternative ways that can happen.
Right. So there's also evidence suggesting that inflammation in the brain can initiate these kinases, right? So going back to the primary tauopathies, frontotemporal dementia, talking about chronic traumatic encephalopathy, you usually see tau proteins that build up there, and there's really no evidence of amyloid beta being present. So yes, the question is, why is that? And what we're finding is that for example, the example of CT is a good one, chronic traumatic encephalopathy, that's typically triggered by repeated head blows, right? Traumatic brain injuries. And we know with those injuries, you typically have inflammation that is elevated in the brain and chronically elevated. So there is evidence actually now showing that having increases in inflammatory molecules in the brain can actually lead to increases in those kinases as well that can then initiate the hyperphosphorylation and those neurofibrillary tau tangles. Yes.
I see. So if we think in like a 2x2 grid here, tau protein present, absent, beta amyloid plaques present, absent. It sounds like in almost all cases of Alzheimer's, when you do the autopsy, you're going to find both tau and plaques, but perhaps in a minority of cases, you find the hyperphosphorylated tau and the tangles without plaques because it was caused by some other thing. And you're essentially never going to find, you might find, an absence of the plaques, but you're never going to find pathology in the absence of tau. And you're usually not going to find it in the absence of plaques, but sometimes you will.
Yeah, I mean, I think that's a nice summary, and that really, highlights the complexity of these diseases, right? That some of them are initiated by an aberrant, protein like amyloid beta, and some of them are by the inflammatory molecules because of repeated head traumas and that sort of thing. But I think the point here that I want to make is that it goes back to the general premise that ultimately the tau tangles is what you really see that drives a lot of these different clinical symptomologies, right? But the evidence that's being put out there now tied to the therapies that actually target amyloid beta, right? Like lecanemab, that's now clinically approved. Their hypothesis is that if we halt the amyloid pathology, that can lead to a halting of the actual tau pathology, right? And there's actually some evidence of that. If you look at a presentation that was given by Biogen in 2024 at the Alzheimer's conference, they actually show that individuals that have been on lecanemab now, this antibody therapy that essentially tags the amyloid beta plaque for degradation, they actually also show some signs of decreases in tau pathology as well, right? In these Alzheimer's cases. Yeah. So I think that points back to the fact that the amyloid beta plaques likely are initiating a cascade of events tied to the tau pathology, whereas you can also get the tau pathology due to other insults like the traumatic brain injuries. Yeah.
Got it. So what seems to be really key here is something, which can often be the beta amyloid plaques, but it could be other things, but something is going to trigger kinases to become more active and hyperphosphorylate the tau protein. That's when you really start to have a problem. Thinking ahead to some of the other stuff that I know that you work on that we're going to talk about, you mentioned traumatic brain injury. You could just get, blunt force trauma, which triggers inflammation that could start to initiate this hyperphosphorylation process independent of, say, beta amyloid plaques. I would imagine that in theory, at least, there could be, say, chemicals or toxins in the environment that might upregulate kinase activity and lead to the same result. What do we know about possible environmental toxins that might tie into Alzheimer's disease?
Yeah, there's a lot of evidence coming out now, looking at various plastics and how plastics that might actually migrate into the gut microbiome. There's a lot of connection between the gut microbiome and the brain. Might actually show up in the brain. These plastics might show up, and we know that anything foreign that has the ability to cross the blood-brain barrier, right? Blood-brain barrier, you can think about it as vasculature that has a variety of barriers preventing things from getting into the brain because what you want to do is you want selective things to get into the brain, chemicals, compounds into the brain, because if they're pathogenic, for example, or they're viruses or pathogens that can actually harm the neuron, and if the neuron is damaged permanently, in many cases, they don't regenerate, right? So what you're finding is that plastics, for example, have the ability to get through this barrier. And there's actually recent papers that are actually showing that it's present in the brain, which is very alarming. In terms of other toxins, we've studied quite a bit the herbicide known as glyphosate, which is a product that is used, a chemical that is used to control unwanted weeds in agricultural communities, right? And the idea with glyphosate is that it's deemed safe to mammalian species because the way it works is by blocking a pathway known as the shikimate pathway. Shikimate pathway is found in plants, and essentially, if you block that pathway, you block the production of important proteins that leads the plant to die, right? But the idea when it was first initiated was that, or when it was first used in the 1970s, was that mammalian species do not have a shikimate pathway, so any glyphosate that you consume is literally going to just be excreted out of your body. But we're finding that that's not the case. As you've probably seen, there's been a lot of research and litigation tied to a lot of effects that glyphosate can have on cancers, right? So a lot of non-Hodgkin lymphomas, a lot of blood cancers, et cetera. Our goal with the research we just recently published in the last couple of years was to understand what are the effects it can have in the brain. And what we're finding is pretty daunting, to say the least.
Yeah. So before we get into your work, I want to build some concepts for people here. So we'll sort of apply some scientific thinking to a topic that sort of bleeds into other issues. So glyphosate, this is an herbicide, as you said, basically a weed killer. This is the Monsanto Roundup product or whatever it's called. It's used all over the place. It's a very, very highly used product. So glyphosate is in the environment to a large degree. I think most people know that at this point. The idea when this thing came out was, okay, well, in order to develop this, they probably figured out, oh, okay, we want a chemical that's going to disrupt some pathway in weeds and plants that's not found in animals and humans. Therefore, it's only going to affect the plants and it's not going to do harm to us. Another way of saying that is if we develop a compound that disrupts a pathway in plants that's not found in animals, and we make the critical assumption that it's not also impacting other pathways in some way, shape, or form in the body, then it would be safe. And I would imagine that the lawyers at these companies probably used arguments of that kind to get it deemed legally safe or safe in a legal sense. But, just thinking sort of, a priori here, what are the odds that a compound like this, even though it operates for this particular application on a plant pathway that's not an animal, what are the odds that it wouldn't have any other effect in an animal body?
Yeah, that's a great question. I would say that based on what we know, based on the literature, I'd say the odds are it probably is affecting, it could affect other specific pathways. And one of them in which we're seeing it's affecting is that it's initiating various aspects of inflammation, right? So inflammation, once again, is tied to many disorders, right? Many diseases, not just, brain diseases, but also peripheral body diseases. And what we're finding is that exposure to this herbicide, glyphosate, is in fact inducing inflammation in the periphery body as well as in the brain. But what's really fascinating as well is that yes, the assumption was made that we couldn't process or metabolize glyphosate, but a paper came out in 2020 that actually showed humans can and mammalian species can. And how is that? The gut microbiome has bacteria. That bacteria has the ability to metabolize glyphosate into what's called amino methylphosphonic acid, which is AMPA. And studies have shown that AMPA itself is pretty toxic to the body. So what we're ultimately finding is that glyphosate does in fact have the ability to affect other signaling pathways in detrimental ways. Yeah.
So let's talk a little bit more about the chemistry of glyphosate. What kind of molecule is this? Is it big? Is it small? Does it resemble anything that's naturally found in the body?
Yeah, it's a small molecule. In terms of its resemblance to anything else in the body, nothing comes to mind. But it's a small molecule that can leak through the blood-brain barrier pretty easy.
Hmm.
And we've seen that it has the ability to do this using cell models as well as in vivo models. Yeah,
Mhm.
in terms of its homology to other endogenous molecules in humans and in mammalian species, nothing comes to mind. Yeah.
Mhm. And so when it was originally deemed safe, I would suppose this is probably back in the 1970s there or thereabouts, when it was deemed safe because it operates, its primary mechanism for its application is through a plant pathway that we don't have. It sounds like no, did no one just check to see if it can be metabolized by the human or get through the blood-brain barrier? Or why did the work that you just cited, why is that so many years in between that work getting published and the introduction of this thing into our environment?
Yeah, I think the answer to that is the way that I would answer that question is that, some of this work was investigated. They would give glyphosate to rodents, for example, and looked at toxicity effects, and they didn't
Mm.
really find a lot of toxicity.
Who's they?
Researchers, scientists, there is evidence. The EPA did some of this as well, and scientists by the EPA, and didn't really identify a lot of toxic effects, right? But they also did different dosages, right? And we do actually have what's called a NOEL, a no aversible adverse effect level that is published actually by the EPA and other organizations as well. At a certain amount or a certain dose, there is actually toxicity that is present. Right. But in terms of, examining whether this actually blocked the shikimate pathway, the answer always was, well, there is no shikimate pathway in mammalian species, so it's not going to be present there. So what do you really look at in that regards, right?
Yeah, yeah.
Yeah.
And I would assume, I'm sort of, I guess, foreshadowing a little bit. I would assume that a critical assumption that goes into that type of work is, well, there's a difference between acute and chronic toxicity, and there
Right.
could be a difference between something being toxic, at a given dose right now that you inject into an animal versus, say, toxic if something accumulates in the body over time. And if the only way to determine if that's true is to do very long-term studies, you might not pick that up in the types of studies that would be done initially.
Absolutely correct. And the thing we have to keep in mind is, in addition, is that these studies are going to be done in rats. They're going to be done
Yeah.
in animals, right? You can't dose a human. That's not allowed. So you do these studies, and even if you did the longest dose, time frame possible, rats and mice don't live, the same time frame that a human does, right? So you're probably talking about rodents 28 months at max. So even if you dose an animal that long, it's still not going to be the same as if you dose a human for 75, 80 years to look at the impact, right? But I will say that some of the work that's coming out that is starting to shed light in this, and this has come out in the last few years, which also made us very interesting, is that if you look at the circulating levels in urine of humans in small sample studies, you find that about 90% of us have glyphosate circulating through our bodies at all times, right?
At
That's
some level.
at some level, correct. Now, what those levels are, they're going to vary. We are actually currently looking at what the differences are, for example, between rural versus urban communities, right?
Yeah.
You would expect in rural communities where there's more agricultural fields and more of this product spray that there's probably going to be higher amounts of glyphosate present, right? But the other component is that, a lot of the foods that are consumed on a daily basis by consumers do have glyphosate present in them as well, coffee and among other items as well. Yeah.
Yeah. So this isn't just found in like a Roundup bottle that you can buy and use in your home backyard. This is used at an industrial scale.
Correct. Yes, it is. So just to give you an idea of the amounts and how widely used glyphosate is, we're talking about 300 million pounds in the United States per year.
Mhm.
Yeah. So it's used very heavily to control unwanted weeds in these agriculture communities, which ultimately, are growing all the product and produce that we as consumers purchase so we can, eat and survive. But that means that, we're being exposed to all these to this specific herbicide. Yeah, because of that. Yeah.
Um, Well, there's a number of ways we can go with this. Let's talk next about what you guys are finding in your lab. So you've done studies where you actually give glyphosate to rodents for
Mhm.
quite a long time, especially relative to their lifespan. You give it to them, you look to see what happens. You also give it to them, and then you stop and you wait to mimic, say, an exposure that might happen for a number of years early in life. And then, someone goes away. Are there long-lasting effects? What are some of the studies that you've done, and why did you? What was the design?
Yeah, so the first study that we really wanted to conduct was back in 2020, really after the groundbreaking work showing that glyphosate can actually be metabolized by mammalian species, and tied to the gut microbiome, metabolism, et cetera. So what we wanted to do is to simply say, if we introduce mice to glyphosate, and these are normal mice, not genetically altered mice, and I'll talk about what those are in the next couple of minutes. But normal mice that were 4 months of age, what happens if we give them glyphosate for 4 weeks?
Yeah.
Do we notice that there's increases in inflammation? We hypothesize that's probably true. But the key component and why we wanted to do this is given that we've established collaboration with one of our neighboring institutions, TGen, one of the researchers there developed a technology where they can actually detect glyphosate and AMPA, the metabolite, in the brain.
Mm-hmm.
That had the capacity to do that. So our goal was to simply say, if we give glyphosate to these rodents, do we see it being present in actual brain tissue? There had been evidence of this in 2019 using cell models in dish that mimicked a blood-brain barrier type of mechanism,
Mm-hmm.
and it found
No
that
one
it
had showed it in vivo, though.
no one had showed it in vivo, correct. And what we actually found is that after the 4 weeks, this was 14 days, excuse me. We actually found that after 14 days, we were able to detect it in the brain. And we also detected the
The
metabolite
metabolite.
AMPA, and we detected in a dose-dependent manner, right? So the more
Yeah. The
glyphosate
more you take, the more you get.
we found. Now, one thing that was criticized because of this work were the dosages, right?
Yeah.
So
Okay.
it's you were probably going to allude to that because that's a question that we get all the
Yeah.
time. And the logic behind those dosages was we were using the NOEL, the no aversible affective level that is currently published as not showing toxicity, which is 500 mg per kg. And then we dose down to a level that's actually the level that is tested and then derive that the human dose is then derived based off, which is 175 milligrams per kilogram.
Yeah.
And
So
yeah,
in theory, so you did, you've got to do conversions and math and stuff to convert, mouse dose to human dose. We won't get into that, but basically what you're saying, if I'm hearing you correctly, is supposedly there's a dose where in theory it's not going to be toxic in humans, and the idea would be like, okay, if people are exposed to a little bit because it's on their produce or whatever, you can go up to this amount and you're probably okay. You basically calculated the equivalent dose and gave that to the rodents.
yeah, it's higher. It's higher than
Okay,
that,
it
right?
was higher than that.
It was higher than that. So I want to make this clear, right? And it's a proof of concept. It's higher than that. So think about it like this. The studies that establish the tolerable amount per day for human, which is 1 milligram per kilogram of body weight, right? That is the
Mm-hmm.
amount that the EPA says you should not go higher than this, was derived by animal studies that gave 175 mg per kg.
Got
Okay.
it.
And then they said, based on what we're seeing in these rodents, we probably should not take more than this, a day to avoid any toxic effects. Our dose that we used in our study, the lowest dose was actually 125 mg per kg, but it's still higher than what the tolerable dose for humans
Yeah.
is recommended by the
Yeah.
EPA. Yeah,
But I think it's worth just making clear for people. So everything you just said, the tolerable dose for human beings is a number derived from
correct.
studies done in rats, and it's we're baking assumptions into that.
Yeah,
So when we say it's tolerable, it's no one has ever shown that the dose tested in humans doesn't lead to any pathology. They've
right.
sort of projected what we imagine might be the tolerable dose from animal studies.
Correct. And also another point that I think is critically important, if you look at Europe and you look at the European recommendations of tolerable doses, their dose is half to what our dose is in the United States. So they recommend no more than zero point 0.5 milligrams per kilograms of glyphosate per day. And why that is, they just said that that's what they believe should be the tolerable upper dose. So even within different countries, we kind of disagree on what those amounts should be. But the point being here, and what's critically important to establish is that the glyphosate has the ability to cross into the brain, which
Yes.
no one had really shown at this phase, and also its metabolite, suggesting that it's being metabolized by mammalian species.
And I mean, one more thing that's important just to put in people's brains, I think, is just because you used a higher dose and you found it in the brain, it doesn't mean that if magically the dose crosses some threshold, it like doesn't get into the brain at all. You're going to expect it to get into the brain in proportion to the dose that gets into your body.
Right.
And so you guys showed, okay, at some dose, it does get into the rodent brain and its metabolite is there. So some kind of metabolism is happening. And I think you mentioned previously that metabolite itself is known to be toxic.
Yes, there's actually studies showing that AMPA itself, in the periphery body, for example, elicits inflammation, and it's very hard to degrade, right? Some of the questions that we got from our second paper, which I'll share next, were, well, why was it still present there? Why was AMPA present? What is it doing? Well, it shouldn't be in the body, and if it shouldn't be in the body, your immune system is going to try to remove it, and if it can't, guess what? You're going to have an increase in inflammatory molecules that can lead to other detrimental outcomes for other tissues.
Yeah, so your body will naturally try to remove things that are foreign to it if it detects them and if it can. And if it can't, you can just get a persistence of these molecules or an accumulation of them, and then that's going to naturally instigate chronic inflammation, which is not going to be good.
Exactly.
And you're detecting, like, it seems like that's what you're detecting in these animal models. You're detecting inflammation.
Yeah, so in the first study, what we did is we looked at pro-inflammatory cytokine TNF alpha. This is known as tumor necrosis factor alpha. Elevations of this pro-inflammatory cytokine have been tied to various cancers, to very detrimental outcomes in the peripheral body. And in the brain, usually those individuals that have neurodegenerative disorders usually see increases in TNF alpha as well. Yeah.
Mm-hmm. So it gets into these mice. The metabolite is there. Can you talk a little bit more about the time course here? Remind us, how long were you giving it to them, and how long was it persisting?
Yeah, so for this first study, we gave it for 14 days. And then after the 14 days, we went ahead and the animals were euthanized. And then we looked at the outcomes based off that. And our second study, that's the study where we actually looked at what are the long-term consequences, which we can get into that. The study was published just December of 2024. And what we did there with our team, we wanted to understand, there's a lot of kind of confusion of, can AMPA and glyphosate, be completely excreted out of your body after a given time, right? And the idea is that, well, glyphosate, since it can't be metabolized, it should just be removed out of your body, excreted out through urine, no problem, right? However, there's evidence now that it can be metabolized by your gut microbiome. You have the AMPA, that's also a problem. So what happens to that AMPA, and what happens to the glyphosate itself in the periphery and in the brain? And what we also wanted to do, extend these findings, is we wanted to see whether in a model that actually has or shows Alzheimer's pathology, amyloid beta plaques and tau tangles, whether the pathology gets worse, right? So these mice are genetically engineered to develop the amyloid beta plaques and the tau pathology. Mice in general, if you just grab a wild type mice or a control mice, they don't develop the pathology that we see in humans, right? So we genetically engineer those mice to develop amyloid beta plaques and tau tangles. So we gave control mice and the Alzheimer's mice, we gave them one of three different dosages. It was either our vehicle dose. We went then down to 50 milligrams per kilograms a day, which is much lower than what our first study, the lower dose was. And then we did 500 milligrams per kilogram as well a day, because that's equivalent to the NOEL, right? The versus effective level. So we dosed them for 13 weeks. And then we said, let's take them off glyphosate completely.
Yep.
And let's see what happens 6 months later.
Yeah. So I guess the loose comparison here would be like, I grew up on a farm. I'm being sprayed with glyphosate for all my childhood, but then I moved far away and I'm not exposed anymore. You're sort of modeling that type of situation.
Yeah, I would say you're moving away, but you're also on a full organic diet, let's say.
Yeah.
Right,
Okay,
where glyphosate is no longer going to be introduced into your system. What did we find? We tested that behaviorally. We did a variety of different behavioral tests. We measured longevity of life. And then we also looked at a variety of neuropathological hallmarks. And then what we did is we did what's called an inflammatory panel. We didn't just look at one molecule.
Whole bunch.
We looked at 23 of them, right?
Yeah.
In the brain and in the blood. And what we found was that while glyphosate itself was no longer present, it was, obviously excreted out, AMPA was in the brain in a dose-dependent manner.
Wow.
So the AMPA made it to the brain, but it wasn't able to be removed from the brain.
And it just persists.
It just persisted there. And if you look at the levels of amyloid beta and tau pathology in the genetically modified mice, you find that their levels go up in a dose-dependent manner.
Hmm.
You also find that the neuroinflammatory signature, the molecules, virtually all the molecules that we looked at were off the charts
Yeah.
in the brain and in the blood.
Yeah. And it really makes really good logical sense. This is a foreign substance. It's not supposed to be there. If it gets into the brain, it's probably just naturally pretty hard to remove things once they get past that barrier. You do find it there. It does persist. And as you would expect,
Yeah.
inflammation goes up.
Yeah, and along those lines, going back to the inflammation, what we found that is even more alarming and convincing of the effects of AMPA eliciting these effects was that the normal mice, the non-transgenic mice, the mice that do not have genetic mutations to develop Alzheimer's pathology, they had elevated inflammation as well in the brain, right?
Yeah.
So a lot of the, criticism as well, the genetically modified mice are probably going to have inflammation because they have amyloid and tau. Yes, they did, but then they had even higher when they were dosed.
Yeah.
But when a normal mouse that doesn't have any genetic modification shows inflammation in the brain, that's indicative of really dire consequences that can actually initiate a lot of the pathologies that we first talked about when we started this conversation. For example, the tau pathology. Yeah.
Yeah. Yeah. A slightly different way of saying all of that would just be that in two different lines of mice that you tested, you found inflammation go up in response to the glyphosate administration.
Which they received 6 months
Yes.
prior, yes.
Yeah. Which is, I guess for a mouse, I can't do the math in my head, but that's like, years of time in a human.
Yes, correct. And the other component tied to that, which I think is critically important for your audience to know, is that most individuals that have glyphosate circulating throughout their body, they don't have it in short periods. They have it throughout their lives,
Yeah.
right?
Yeah.
Granted, the dosages vary, right? Because depending on how much, of glyphosate they're actually consuming based on the products they're consuming, varies. However, the point is that most Americans have glyphosate circulating
Yeah.
in their body throughout their life. So even a short burst of this or
Yes.
exposure to this can have some effects
Right.
to, yeah.
Right. If it's true that it persists in the human brain as it does in the mouse, and you didn't look at humans, but you would imagine this is a very strong possibility. Even if you get a quote unquote short exposure or a low dose, if that happens recurrently throughout time, or it happens, for a year or two, only, so to speak, this metabolite could persist for many years after that.
Right. Yes, absolutely correct. And also, just to highlight another paper that came out a few years ago, they examined the levels of glyphosate in urine in a cohort of individuals that are known as a study called Anne Haynes study. Right. So a group has a group of elderly individuals that they follow and they measure a variety of different metrics. One of the things they wanted to measure was glyphosate in their urine. So they measured glyphosate in their urine. They found that individuals that are older had slightly elevated glyphosate in their urine. And what they then measured was cognition, clinical outcomes, right? Now, these individuals were not Alzheimer's disease patients. They were normal, cognitively aged individuals. But what they found is that as the levels of glyphosate in your urine go up, your cognitive scores went down. Right. Showing that, highlighting evidence that's consistent with our work, showing that behavioral outcomes can be affected by glyphosate levels in your body. And furthermore, that same group published a paper showing that the levels of glyphosate correlate with a biomarker known as NFL. NFL is known as neurofilament light chain, right? And it's actually used as a biomarker to understand the progression or to predict the progression of Alzheimer's disease. And what you find similarly with the clinical score, with the clinical performance, that is glyphosate levels went up in the urine, so did the NFL. NFL, when it goes up, it's indicative that there's neuronal damage occurring in the brain.
Yeah.
Yeah.
Yeah. So, I mean, it all fits. Obviously, we can't go and take people out in the world and look in their brains like you can on the mice. So, people might say, well, that's just correlation, but, there's no way we can really dig into it. It all fits with the work that you showed in the rodents. And it just makes sense. Like, what are the odds that glyphosate is going to be totally benign?
Yeah.
It seems unlikely.
And I think one of the things that we are now focused on, because this is, as I mentioned, criticisms that we usually get with this work is, well, your dosages are slightly higher than the actual human exposure doses. We're doing this now. We
Yeah.
actually took rodents and we're testing this at the actual human allowable doses of the 1 mg per kg, and we're seeing what the effects are there. We're also comparing that to the zero point 0.5 mg per kg that is currently being, that's currently the limit that you
In
see
Europe.
in Europe, and then also doing a dose slightly higher because there's actually evidence that's been published showing that since rodents have a faster metabolism,
Yeah.
dosages need to be corrected. And what
Right.
is the value? You multiply whatever the dose is by 12.3. So we have rodents that are getting 12.3 mg per kg. That'll tell us, based on this, is this amount, which is synonymous to what humans should, be at the limit? Are we seeing these effects there as well? Yeah.
Yeah. Wow. Um, well, I don't want to jump ahead and make assumptions, but I'll be shocked if you don't see something. Um, okay. Well, yeah, that's all interesting / frightening stuff. Um, so the idea here is glyphosate can in fact be metabolized perhaps by our gut microbes. This metabolite AMPA is itself toxic. You found that persisting in the brains of mice. Pretty good chance that's happening in humans as well. If it persists in the brain, that means that the brain has no endogenous way of getting it out of there that works very well. Is there any way to obviously, you can reduce exposure to glyphosate by avoiding things that are likely to have it. Is there anything known about how to lower, say, glyphosate that's in the body? Is there anything that leaches it out of your blood? Do we know anything there about how people can maybe reduce what's inside of them already?
Yeah, that's something that our collaborator at TGen here in Arizona is trying to look at. He's trying to look at whether there's ways to potentially tag it and just, allow it to actually be removed. But once again, as I said, glyphosate isn't really. I mean, glyphosate is ultimately what we are consuming and what's causing the AMPA. But really, the question that we're going to ask next, and we're trying to investigate, is how do we actually remove the AMPA? Because that's what we're seeing, that's present throughout this what we call recovery period, where the animals were not exposed to any glyphosate. So we're trying to work with many brilliant chemists and biochemists because it is an area that they have better expertise in than I do, and we bring in the cell biology component. But that is definitely something that we're trying to see. One, why is it that the AMPA was not able to be removed from the brain? And two, are there ways that we can actually allow that to occur? So it's in the beginning phases, and I would say stay tuned because sure, hopefully we'll have something that allows us to test that. Yeah.
And what can you tell us about how to minimize exposure? Is it true that if produce, say, is organic, that it is likely to have much lower levels of glyphosate on it? How do people find things that are less likely to have glyphosate?
Yeah, typically organic foods are going to use less of the herbicides and pesticides, right? So typically you do find individuals that eat full organic diets have lower levels. Although I do want to warn that there is evidence that some individuals still use aspects of glyphosate even for organic farms, et cetera, right? But typically that's one way in terms of, really moving towards a organic diet. Although that brings in a very complicated component, and that is, organic diets aren't cheap, right? And who is going to have access to this? So it's complicated, right? I will say the least, because most of the individuals that read our work and ask the question say, well, if we can't use glyphosate, what are we going to do with unwanted weeds, and how are we going to get products to the consumer that's going to be affordable? So it's a very complex problem. It's not just about let's reduce glyphosate, but how do we also make it affordable for everyone to be able to eat?
Yeah. Well, speaking of eating, I want to talk about how nutrition impacts neurodegenerative disease, how maybe neurodegenerative disease can be caused by nutritional deficiencies and things like that, or sort of the flip side of that, which is are there nutritional interventions that might slow down or reverse or help prevent things like Alzheimer's? And I think you've done some work there as well. Maybe you can tell us where a good place to start is.
Yeah, so I mean, this is one of my favorite topics to discuss. I've been working on the nutritional aspects tied to neurodevelopmental and neurodegenerative diseases now for about 15 years. Started this work as a graduate student at Cornell University, and what we know about the role of diet in the prevention of disorders like Alzheimer's disease is that there's a lot of data showing that the MIND diet, right? So this is known as a Mediterranean DASH diet, which is very heavy on berries, poultry, fish, for example, that have a lot of nutrients that are critically important for our body, right? Antioxidants, for example. There's a lot of actual clinical trials and evidence showing that that is beneficial for an individual in terms of their brain health and deterring from disorders like Alzheimer's disease. So the Mediterranean DASH diet, also known as the MIND diet, has a lot of efficacy in that regard. Now, tied to the work that we do, we focused primarily on what's known as choline. So choline is a B-like vitamin nutrient that is critically important for a variety of body and brain related functions, right? You can produce choline endogenously through your liver. However, studies have shown that it's not enough for the roles that it plays in your body, right? And so
Mhm.
we could touch on what those specific roles are. So one, it plays a role in fat metabolism. If you don't eat sufficient choline per day, you tend to see that individuals develop fatty liver disease,
Hmm.
right? That's in fact why the Institute of Medicine in 1998 first made the recommendations for choline for men and women, adult men and women, which are currently for adult women they're 425 milligrams per day, and for men 550 milligrams per day. Now, there's also a recommendation for pregnant women because choline is critically important in building phospholipids, which is essentially what builds our cell membranes, right? That
Mhm.
protects things from going in and out of our cells. Women are required to consume 550 milligrams of choline per day, and not doing so has actually shown to potentially cause neurotube defects and neurodevelopmental delays and disorders as
Mhm.
well.
Mhm.
Okay.
So one of the main areas of choline's functional effects is fat metabolism, broadly speaking, all different aspects of what the body does with fats.
Yes, yes, it is. It allows for the processing of fats. It works closely with, once again, your liver. And it also intriguingly plays a role in regulation of insulin, right?
Hmm.
So, as we know, diabetes is a big problem in the United States and really the world. And what we're finding is that that fat that accumulates in your body because of lack of choline can actually contribute to insulin resistance, which is a feature of type 2 diabetes. Now, if you look at the cases of type 2 diabetes, those that have it, the percentage of individuals with that disorder that develop Alzheimer's disease is really high, close to
Mm.
80%, right? So it's all actually linked back
Yeah.
to metabolic function and fat metabolism, right? Having improper ability to reduce fatty liver disease can then trigger a cascade of events throughout your body that can then predispose the individual to actually develop disorders like Alzheimer's disease.
Yeah. Yeah, that's interesting because I'm hearing more and more people talk about Alzheimer's as type 3 diabetes. Maybe we'll loop back to that, but
Sure.
with choline, um, before diving into some of the details here, where do you normally find it? What foods are abundant in choline?
Yeah, very good question. So the one of the top one of the foods where you find choline in high concentration is going to be eggs, right?
Mm.
So eggs have a very, very high concentration of choline. If you eat 3 eggs, you're pretty much hitting the recommended amount that you need per day. It's found in poultry, it's found in fish. It is found in red meats, leafy greens, and grains. Now, as you recall, I talked about the mind diet early on, right? Where I said the mind diet that requires poultry and fish, et cetera. All those components that make up the mind diet are also very, very rich in choline, right?
Mhm.
So the idea is that that diet plays a very important role because it not only has other forms of omega-3s, for examples, and antioxidants, but it also is enriched by choline.
Yes, and an important thing to point people to here, if you can shed any light on this, please do. But as with pretty much any nutrient, there is usually there's a key difference between the nutrient content of a food, how much is inside of it, and the bioavailability of that nutrition. And I would imagine, without knowing the specifics myself about choline in particular, that the nutrient bioavailability is going to differ quite a bit between some of those different foods that you mentioned.
Yeah, they do differ. As I mentioned, in eggs, you typically have more choline that's going to be present. In terms of the poultry and the fish, you usually find high concentrations there as well. With some of the leafy greens, you do have it, but it's not available in really high concentrations. And there is then an issue that actually I wrote an editorial on that I think is really important to point out. What happens to, for example, plant-based eaters, right? So
Mm.
plant-based eaters don't have access to fish, poultry because they don't consume those products. Does that mean that they're more likely to be deficient in choline? And the answer to that is yes. But you still have the ability to buy over-the-counter supplements for choline, and they're pretty affordable. That allows you to reach the level of choline that you need if you're a plant-based eater by taking those extra supplements as well. And they vary drastically. So I have a lot of individuals that reach out to me and say, "Hey, I'm taking this multivitamin and it has choline. Am I okay?" And a lot of times I say, "No, you're not," because multivitamins usually have 15 milligrams of choline, maybe 20 milligrams of choline.
You want
So
hundreds per day.
yeah, so you need hundreds, right? So if you are deficient or are eating mainly a plant-based diet, it is recommended that you actually get a bottle that has actual choline in it in the
Mhm.
hundreds of
Mhm.
milligrams per day.
Um, yeah, and I'll just ask you this real quick because I think it's an interesting and important question. This is not the first time I've heard of this type of thing, where if you're eating a primarily plant-based diet, it's harder to get enough of, in this case, choline in your diet to hit your minimum requirements. That's true of various other nutrients as well. And yet, if I go see a doctor, the last time I went to a physician here in my home in Seattle, and I hear this from all sorts of major health institutions, they promote primarily a plant-based diet. Is it strange to you at all that plant-based diets are really in vogue right now with major health organizations, and yet they're often low in bioavailable nutrition like choline?
Yeah, I think what that is, it points to aspects of, the literature suggesting that if you eat red meats, if you eat a lot of these other products, eggs, for example, that might spike, cholesterol, for example. We know cholesterol can be risk factors for heart attacks and other potential ailments that you find throughout your body and your brain, right? But I think what we're doing in our lab and really the community that is looking at a variety of these nutrients that are really needed is raising awareness, right? So if you look at the amount of individuals that actually know that they need choline, I do this every time I give a lecture. I always say, how many of you know what the recommendations for choline are? Usually you get two or three people out of a crowd of,
Mm-hmm.
50,
Yeah.
100. So 90% of Americans are currently not getting the adequate amount of choline that they need.
90.
90%, right?
Yeah. Yeah.
They're getting approximately 50% or even lower than that, right?
Yeah. And
So
it's,
most
I imagine a lot of them don't even know that choline is a thing. It's not on the nutrition labels or anything.
Exactly. And, I was having a conversation with my younger sister recently because she's followed my work since I was a grad student and especially tied to aspects of pregnancy. And she's pregnant and she asked me, "Hey, I'm pregnant and I've read all your literature, choline. How much should I take? Should I discuss this with my doctor?" And she did. And her doctor said, "Well, we just focus on folic acid. If you want to take choline, that's kind of on your end. But we know that it's recommended by the Institute of Medicine and it has been since 1998. Yet, it's not being pushed as much compared to other aspects of the nutritional realm in pregnant women. So part of what we're doing is also trying to educate physicians and other medical professionals to understand that this is critically important based on all the literature that we're publishing and putting out there.
Okay. Well, choline's important. I'm definitely getting enough choline because I eat a lot of eggs, I would imagine.
Very good.
So choline reminds me of acetylcholine, which is a neurotransmitter in the brain. So why don't we talk about the role of choline in brain stuff now? How is it tied to brain function in basic terms?
Yes, so choline is essentially the precursor to acetylcholine. What is acetylcholine? Acetylcholine is a neurotransmitter. You can think about it as a chemical messenger signal. What it does, it mediates a lot of behaviors. It is released in synapses that allow neurons to communicate, and it produces a lot of important functions for us. So, for example, learning and memory relies on acetylcholine. If you don't have adequate acetylcholine, thereby not enough choline, that's gonna, that behavior is gonna be affected. It's also important in what's called the neuromuscular junction. So you can think about the projections from your spinal cord that are part of your central nervous system that are going to synapse to skeletal muscles that allow us to move our muscles, right? Acetylcholine allows that to happen, right? So if you're deficient in acetylcholine, a lot of these behaviors are going to become compromised. And what we see in Alzheimer's disease is that acetylcholine is one of the first signs. The drop in acetylcholine is typically what you see in many cases of Alzheimer's disease because the neurons that are actually producing this are compromised and begin to die. Right. And we also find that individuals that eat less choline have an increased risk to developing Alzheimer's disease. There's been a few papers that have been published in the last 2 years, one coming from my lab that actually shows that decreases in choline can actually lead to decrease in acetylcholine, which can predispose a person to develop disorders like Alzheimer's disease.
Is there any evidence that like supplementing or getting, is there any evidence that nutrient supplementation or just ensuring that you're starting to get adequate levels of things like choline can like reverse problems like neurodegenerative problems?
Yeah, so there has been a trial that was done many years ago where they actually supplemented with a form of choline, phosphatidylcholine, and they didn't find benefits in a clinical trial. However, I will say that was a study that was done in patients that were more in advanced stages of Alzheimer's disease. So can it reduce the plaques, the tau tangles? No. If they're there, they're there, and the choline is not going to remove that, right? However, what we have found based on the literature is that if you start early, if you get your recommended daily intake amount, in your 20s, 30s, 40s, and beyond, you're essentially preventing aspects of the disease that are risk factors and can initiate the cascade of pathological burden that you see in these specific disorders. So in my lab, we've published a few papers in rodent models actually showing two phenomena. One, if you don't have choline in your diet, it increases, it leads to obesity, it leads to liver pathology, it increases plaques and tangles in the brain and neuroinflammation. That's if you don't have enough choline, right? Or don't have any choline in your diet. The opposite is true. If you supplement with more choline, you actually reduce the amyloid beta and the pathology that you see in Alzheimer's disease. However, it has to start pretty early.
Yeah.
If you try to use it as an intervention where the pathology has already began, it's kind of like it's too late, right? It might help. It might help with aspects of metabolic function. If someone has diabetes, we actually are working on a study right now where we're touching on aspects of that, and we can get back to that. But the evidence does show that it needs to be done throughout your life to truly have profound benefits in the prevention and also in healthy cognitive aging.
Mm-hmm. Yeah. So the name of the game here is prevention, and that makes sense. Like from a nutritional standpoint, if you're getting adequate nutrition, your body will be able to do the things it needs to do and be less likely to develop pathologies. But at least in the case of something like Alzheimer's, when you've got tangled microtubules and proteins in your brain, you're not going to get rid of those most likely if you simply start taking something that you're already deficient in for a long time.
Exactly, 100% correct. So I'm a big advocate, and I share this with everyone when I give these talks, that this is established and recommended by the Institute of Medicine since 1998 when they first established that it contributes to fatty liver disease. So everyone should be taking it, right? It's not going to hurt you. I've had questions about, hey, what happens if I take too much choline?
Right.
You need to take approximately 8 times the recommended daily intake amount to have toxic effects. The 2 toxic effects are typically low blood pressure, and you start to smell like a fish because of the metabolite that occurs. Yeah. I mean, not very bad side effects, but...
hey, that at least tells you, that's a clear marker of like, oh, hey, I can back off the choline.
Yeah, but getting close to 4 grams of choline a day
That's a lot.
That would be very difficult to do, right?
Wow.
most supplements and tablets that are out there usually give you about 250 milligrams of choline. Yeah.
Yeah. So you can literally target the recommended dosage in the 400 or 500 milligram range or so, and then you could just go above that to be safe a little bit, and you'll be fine.
Right. Yeah. And we've actually done a few studies that have tested. This is in rodents, but there are studies that have been done in humans, and I'll talk a little bit about those as well. That we typically go at 4.5 times the recommended daily intake amount, right? Which is still not very close to the 8 times, right? And we find pretty drastic benefits in terms of cognition, reductions of neuroinflammation, reductions. Actually, well, one of the big things that we're finding now is that it can actually regulate weight, right? So we actually found that mice that were on a choline deficient diet gained about 30% more weight than animals that had choline.
Really? Same
Right. Yeah.
exact diet, just choline.
Yes. This is published in 2023 in Aging Cell, and it's you can look it up. Choline deficiency promotes a lot of system-wide pathologies and dysregulation of metabolic function. And we found that when we looked at the mice, we were pretty shocked. The only thing we did is take choline away from their diet, and they gained tremendous amount of weight. Yeah,
it is a very basic way to think about this. So why would it have effects on weight and inflammation in the brain and insulin? Like all of these things is a way to think about this simply that choline is a precursor to probably multiple key signaling molecules that have a variety of roles in metabolism and various aspects of biology. And therefore, if you're deficient in it, it's just going to sort of disrupt signaling in lots of different ways.
Yes, that's exactly the way to think about it, right? And it's probably going to do it not in a way where you think where it's like, oh, you're probably going to get, there's going to be obesity, and then you're going to get aspects of diabetes or glucose metabolism impairments, and then Alzheimer's disease or pathologies that can initiate that. They're all probably happening synergistically, and then ultimately you see all of these pop up at the same time. But the one thing that I just want to make this comment. The one thing that we're very excited about is that we're really starting to show that in human cases, individuals that are pre-diabetic have low circulating choline in their blood, right? So if you take individuals that have a BMI of obese, and you compare those individuals to individuals that are normal, healthy weight individuals, you find that the levels of choline in their blood in the obese individuals are drastically lower. Why is that exciting? Because if we can address aspects of prediabetes by simply supplementing with choline, that would be fantastic.
Mm-hmm. And so some of the results that you were pointing to that you found in rodents, to what extent are those results that you're rescuing choline deficiency in mice that are deficient versus can you take normal mice eating sort of standard mouse chow and then supplement with extra choline and then see improvements in anything?
Yeah, actually, that was the premise of a lot of this work, right? So once again, some of the first reports showing that choline supplementation was beneficial comes back from studies done, back in the late 1990s and early 2000s, where they actually took normal rats and they just gave them more choline versus normal choline. And they actually found that having more choline actually improved their behaviors in terms of learning and memory and other forms of cognition. That actually established that choline is critically important and more of it could be beneficial. What we're trying to do with our work is to say, is a deficiency in it, which is more prevalent in our society, potentially leading to a lot of these detrimental outcomes that we see in disease.
And so everything points to, obviously, deficiency is very bad. You definitely want to avoid choline deficiency. Supplementation is good. Even liberal supplementation can have benefits in non-deficient animals, and you're going to be pretty hard pressed to overdose on something like this.
Yes. Yeah. I mean, like I said, getting close to 3-4 grams of choline a day would be very difficult, especially since most of us are deficient, right? Most people are deficient. I myself, my whole lab, we are big proponents of taking choline on a daily basis, given our literature. So we have to practice what we preach.
Interesting. Yeah. And we talked about Alzheimer's earlier. You talked about some of the effects choline has on metabolism, things like insulin resistance. How has the thinking started to change around Alzheimer's and whether or not it's rooted in things like insulin resistance and a metabolic issue?
Yeah. So, going to the evidence, as you mentioned, type 3 diabetes, right? The individuals that are typically an obese BMI, as well as have forms of type 2 diabetes, typically have an increased risk of developing Alzheimer's disease. And the reason why we believe there's a link is because insulin resistance can happen in the periphery, but it can also happen in your brain, right? If your receptors in your brain that require binding of insulin to allow glucose to come in are now compromised, that is going to start to affect the function of those neurons. And studies have shown that if you don't have proper glucose in your brain, that can lead to neuroinflammation. And what does that neuroinflammation tie back to? A lot of these proteinopathies that we discussed earlier in this conversation. Yeah.
So, what are some of the things that you guys are working on in the lab now, either extensions of the work that we've already discussed or interesting areas that we didn't touch on?
Yeah, so a few projects, and both in the human space using human participants as well as postmortem brain tissue and animal models as well. So in terms of glyphosate, as I mentioned, we are interested in looking at the effects of the upper limit of the dose that's allowed to be consumed by humans on a daily basis, at 1.0 milligrams per kilogram per day, the European dose, and then a corrected dose by the FDA in rodents. So we're looking at some of those studies. For choline, we are actually now starting to look at what the benefits of supplementation are for tauopathies, right? So we know there's evidence suggesting that if you supplement the diet in an Alzheimer's model, you do see benefits. What about a disease state where there's only tau pathology, right? So we currently have a few studies where we're looking at genetically modified mice that develop tau pathology to see if it can reduce or prevent some of the aspects of neuroinflammation that, will aggravate that specific type of pathology. Yeah, sorry.
No, go ahead.
Yeah, and another important area that I'm really interested in, it's a fascinating, really cool area of research, is one of our collaborators at, I keep mentioning TGen, the Translational Genomics Research Institute in Arizona. One of our collaborators developed a lab that's on wheels. It's called the Mobile Lab, right? It's very cool. I remember when this colleague came to me and said, Ramon, I have this great idea to develop this mobile lab. We can go around the state or really, the United States and collect data, collect, blood specimens. We just park it and tell people that, hey, come be part of our study. So that's actually now occurring. And here in Arizona, and what we're really doing is leveraging that mobile lab to develop a few experiments tied to glyphosate and also choline. So what we find is that depending on where individuals live, rural versus urban, we're finding that individuals that live in rural communities have a higher incidence of cognitive decline and Alzheimer's disease. Why is that? So we're teaming up with this group and we're going to these different locations and we're trying to understand, what does the circulating choline levels look like in someone who lives in a rural community versus an urban community? What are the levels of glyphosate looking like? If you live in an agriculture community, are you actually finding that there's more glyphosate and also less choline being consumed that might be triggering a lot of these cognitive decline and neurodegenerative disorders that you see in these individuals? Yeah.
A lot of stuff ties together at the level of diet in terms of the dietary choices people make. So, for example, based on the glyphosate story you told me and the choline stuff you told me, if someone was eating a largely plant-based diet with little to no animal products and they were not being careful to A, supplement with things like choline, and B, focus on organic foods, then they're probably getting exposed to more glyphosate and they're probably likely deficient in choline. So these things can, even if there's no direct connection between those two threads, they can come together in a package, so to speak.
Yeah. And I mean, that ties back to these neurodegenerative disorders. They're so complex, right? Yeah. There's no, one environmental factor that we know of that leads to Alzheimer's disease or these detrimental outcomes. It's going to be a combination of the environment, right? Yeah. So our goal is to go and look at what are these different components and what can we do to improve the lives of these individuals, because we know that life expectancy is only going up. More cases of Alzheimer's disease are occurring. What can we do to give everyone the best chance possible to not go through this type of disorder in their life? Yeah.
Well, this is really good stuff. I talk a lot about nutrition, but this is one of the more clear-cut cases where supplementation pretty much makes sense across the board. I'm talking about choline, and deficiency is it's very obvious why you don't want to be deficient in this case. So really interesting, especially if you are eating certain diets, they're going to be naturally choline deficient because you're not eating animal products. So that's really interesting stuff. Any final thoughts you want to leave people with about anything we discussed today?
Yeah, I mean, our goal really is to find ways in which we can improve the lives of individuals that are aging and, the general public. And we believe that, these lifestyle factors play a very big role in, what we will experience later on in our life. And so here at ASU and with our collaborating partners, we're doing everything we can to ensure individuals as they age with their memories and have a graceful aging process. Yeah.
All right. Well, Dr. Ramon Velazquez, thank you very much for your time.
Thank you very much for having me.